Hello everyone, my name is Arteaga and I'm going to present the post characterization of gene regulatory and interaction networks across cell types during SARS-CoV-2 infection using single-cell RNA sequencing data. In this work, we collected 3 datasets that were publicly available that contain single-cell RNA sequencing data from COVID-19 patients and controls. Today I'm going to show you the results from the 2 datasets that are highlighted. First, we collected datasets and obtained gene expression matrices and metadata, and we performed differential expression analysis, gene set enrichment analysis, and we inferred the interaction networks using CellPhoneDB. We inferred the gene regulatory networks using PySynic, and with the results of PySynic, we selected differentially active regulons for every cell type in the datasets. And we performed gene ontology analysis using the differentially active regulons, and gene set enrichment analysis show enrichment of interferon response, inflammatory response, and complement system pathways in both BALF and PBMCs. And we see that interferon response pathways are enriched in every cell type in both tissues. Here we see a heat map of activity of different transcription factors that were differentially active in COVID-19 samples from the scenic analysis. We see that in BALF, macrophages and T cells are gaining more transcription factor activity than the rest of the cell types, whereas in PBMCs, almost every cell type is gaining more activity almost at the same level. We performed gene ontology analysis using the transcription factors that we saw in the previous slide, and we see that in BALF we have enrichment of immune response pathways, and also we see enrichment of RNA metabolic processes. In PBMCs we see that 2 things that are interesting, and those are that B cells, plasma cells, and T cells are not having an enrichment as high as the other cell types, and also that megakaryocytes are having an enrichment of different RNA and metabolic process pathways. When we performed the interaction network analysis, we saw that almost every cell type is gaining interactions with the rest of the cell types in BALF as well as in PBMCs. We also have increased ligand receptor interactions between CD74 and APP, COPA, and MIF, and we have a great increase in COVID-19 samples of these interactions. And this is interesting because CD74 is known to have antiviral activity in coronavirus. We also have increased ligand receptor interactions between TNF, RSF1B, and GRN, and between TFSF13B and HLA-DBP1. These tumor necrosis factors have been observed to have increased activity in COVID-19 samples in other studies, as well as the HLA-DBP1 protein. So we have seen increased activity of transcription factors associated with immune response and relation of RNA metabolism, and increased interaction between proteins involved in immune response, some that have already been described in other studies and others that are novel ones. We continue working on this project and we're still analyzing the results, and we hope to discover more insights into how the host modulates its gene regulatory networks and interaction networks when infected by SARS-CoV-2. Thank you very much.